Welcome to this video about immunity, and we're going to be thinking in particular about the adaptive immunity. So that's the part of the human immune system that responds in a specific manner dedicated to the shape of the antigen that is attacking the immune system. Now, by no means think that this is the beginning and end of this topic. It is incredibly complicated, and we are going to simplify it and just concentrate on the key principles that allow our body to respond in a specific and measured way to a specific antigen, and to remember that antigen in the future so that when the body responds for a second time, it will respond faster and harder than the first time and hopefully prevent us getting sick. So the success criteria, we're going to look at the different types of lymphocyte, the T cells and the B cells that are in the acquired or the adaptive immunity immune system, how antibodies can be used, and we're going to use a model of flow diagrams to show how these things are connected. So let's start off with our overview. So we'll begin and end with this. And if you start up in the top left-hand corner, we've got our pathogens. So a couple of different shapes here of bacteria and viruses that have entered our body. And what we know so far is that the innate immunity, so this is the part of our immune system that exists anyway, and it doesn't really care what the foreign particle is, doesn't worry about the shape, it's just going to attack anything foreign. So macrophages and neutrophils, they are phagocytes, and they will engulf the pathogen and chomp it up with digestive enzymes and present it on their surface when they've done that. Natural killer cells will attack any cells that are expressing foreign proteins on their surface. And there's a cell called a dendritic cell or a dendrite, and it will express antigens on its surface too. So the innate immunity picks up anything foreign and expresses it to the rest of the immune system. And so this is our link to the specific immune system. These cells will move into the lymph nodes, and in the lymph nodes live lymphocytes, white blood cells. And if there is a lymphocyte living in there that recognizes the antigen that has been seen in the body so far, they will get activated. So the first cell to get activated will be the helper T cell. It then speaks to a whole load of other white blood cells, like B cells that make antibodies, and T cells that go around killing specific cells that have been invaded by a virus. And you can see we're linking back having a specific attack on our pathogen. So either by cellular attack or by antibody attack. These little Y shapes are antibodies. So this is what we're going to come back to at the end. We're going to unpack each of these steps. So when we're thinking about the adaptive immune response, we're thinking about certain things that are different from what we've met so far. It is very specific to the shape that's being that's invaded our body. It involves a whole diverse range of responses. So you've already seen it's got antibodies as well as cells. It has memory. It can remember an attack, and it will not attack self. It does not attack your own body cells that should normally be there. And it involves white blood cells whose special name is lymphocytes, and we have two types, T cells and B cells, and the T and B stand for where they have been matured. So they're all made in the bone marrow, but they're mature in different places. T cells in the thymus up here, and B cells in the bone marrow. So when we are activating our specific immune response, our adaptive immune response, the first step is to present antigens on the surface of an antigen-presenting cell. So you can see here phagocytosis has taken place in this macrophage. So we've got a bacteria, and it's been engulfed and surrounded by cytoplasm. Lysosomes have come in and fused with it, broken it up, and there are little molecules called MHC molecules, MHC2, don't worry about the 2, and they have attached little pieces of antigen to them and put them on their surface. And the other white blood cells are going to notice that and come and interact with it. So let's have a look at what happens next. Here is our phagocyte, just a slightly different image. Here is the antigen that has been chomped up, and it is now on the surface of the cell, attached to the MHC molecule there. That stands for major histocompatibility. That's one of those molecules we need to be really careful that we match when we're doing organ transfer. And on this cell here, this is a T helper cell, so T with a little H next to it, T helper cell. That's the first cell that's going to be activated in our system of making sure that we are activating a specific cell. Now, all different T helper cells exist in your lymph nodes. This cell has travelled off to the lymph node, trying to find a T helper cell that happens to match the antigen that it has found. And each of the T helper cells has a different receptor on its surface. It's called a T cell receptor, or TCR. And if that T cell receptor happens to match the antigen here that is presented with the MHC receptor on the surface of the antigen presenting cell, that T helper cell gets ever so excited. It gets activated. And activation, what does that mean? It actually means that it starts to make its own chemicals, telling other cells that it's been activated, and it starts to reproduce by mitosis a lot. So we have clonal expansion and release of chemicals. Let's see what effect those chemicals have. So here is a slightly different image. We have our antigen presenting cell. Let's just see what we're doing here. We've got our antigen presenting cell, so our macrophage, or similar. Here is our T helper cell that has got ever so excited because its T cell receptor happens to match the antigen and the MHC receptor that's presenting the antigen. And we have clonal selection. So we have a clone, it has reproduced by mitosis, and we've got lots and lots of T helper cells here in this row that are all the same genetically, and they can all recognize the same antigen. A few of those T helper cells will go off to a lymph node and basically sit there, twiddling their thumbs, waiting for another time when you meet this antigen. So they become the memory cells. But the other T cells over here, or these guys, these three, they can do several things, and it kind of depends what chemicals they release. Some of them go off to activate B cells. B cells make antibodies. Some of the T helper cells go and interact with other T cells. And there's a type of T cell called a killer cell, which is a pretty cool name, sometimes called a cytotoxic cell, and it will go and kill virus-infected cells, for example. And some of the T helper cells go back to the innate immunity, and they pump up the macrophages and the neutrophils and get them phagocytosing even more than they were before. So you've got a whole boosting of different areas of the immune system, and you've laid down some memory for the future. So let's have a look at the different types of cells that can become activated. So we mentioned all of these so far. We've got killer cells, sometimes known as cytotoxic cells. They destroy pathogens and infected cells. We can clonally expand even more helper cells that just go back and keep activating other cells. We have made some memory cells, but there's also a slowing down option in our immune system, because if it just keeps getting ramped up and ramped up and ramped up, we could end up in a situation where we have an overactive immune system and we start attacking the wrong things. So we have suppressor cells that just calm everything down. You can see in this little cartoon it said, whoa, B cell, just calm down. The infection's going away now. It's okay. Other activated cells are B cells. B cells can become two things: plasma cells that are releasing antibodies, and memory cells, so that we can remember to release the right antibodies in the future. So let's concentrate a little bit on the B cells. So here are our B cells. This particular B cell has become activated. It's got red on it. The right T cell, T helper cell, joined to it and woke it up and said, yeah, your antibody is perfect in shape for this pathogen. Divide and form clones and clones and clones. So you get clones dividing by mitosis, and you get lots of activated B cells. I've just shown two here. they now have a choice. A few of them become memory cells. So they will remember that shape of pathogen for the future, ready to go. But most of them become plasma cells. So they become cells with the ability to make lots and lots and lots of protein. And the protein that they are making are these antibodies here. So antibodies are Y-shaped molecules. So here are some antibodies. They're shaped like a Y. And the key thing about antibodies is that up the top here, they have a specific shape that has been selected for this particular antigen. It matches perfectly, a little bit like when we used to study enzymes and we talked about lock and key. So a perfect fit, and they can now do quite a few different things. Antibodies might neutralize toxins and things. So the first thing that antibodies can do is to neutralize. That means that they just bind to the bacteria or to the toxin and stop it, or the virus, and just stop it being able to do what it wants to do. It can agglutinate. That means clump together. So all the bacteria, instead of being floating away freely, they're now in one big clump. That reduces their ability to invade. Sometimes antibodies bind and then they trigger a whole load of proteins whose job it is to punch holes in the bacteria and cause them to burst. And then there's this final thing called opsonization. And what that really means is to make the bacteria incredibly tasty to phagocytes. So you can see this macrophage here is wanting to eat that bacteria more so because it's coated in antibodies. So those are the things that antibodies can do. There are actually different types of antibodies. We don't need to remember all of these, but it's worth remembering that that Y shape can be put into different configurations. So you can see sometimes you've got two Y shapes together, so like with IgA, and you tend to find IgA in secretions. So you've got two Y shapes, one against another. IgG is the one that gets made after a vaccination. IgM is usually the first antibody you make. You can see that it's got about 5 antibody molecules all surrounding. So there are different types of patterns that you can get, but they still all do the same job. So the final thing we need to think about is the impact of memory cells for the B cells. And we've seen the first part of this diagram already. And here is where the memory cell comes in. Lots of time later, you get the same antigen suddenly in your system because you come across the same disease. And very quickly, the body will make even more memory cells, and incredibly quickly they make lots and lots of antibodies. So you don't even notice that you've got the disease when you have it for a second time. So what you're getting this time is a response that's really fast and really intense. And you'll actually notice it's the different type of antibodies. You get IgG. So the first time you manage to combat the disease, but then after a while, when the second disease comes, second time you get exposed to it, you get a much bigger response, and it's really quick. So that's an advantage to having been exposed before, and that's the basis that we'll talk about when we look at vaccination. So let's go back to our very first flow diagram. It should make a lot of sense now. We've got our pathogens. The pathogens are noticed by cells that just pick up foreign stuff. They don't really care what it is. But they are the cells that are going to present those antigens to the specific immunity, in particular the T helper cell. The T helper cell is our key. It activates killer cells, it activates antibody-producing cells, the B cells, and then we can launch an attack. Humoral attack via antibodies, or a cell-mediated attack via T cells. And that is the specific immune response.